Hey everyone, welcome back. So in my previous video, that is part 1 of this amino acid video, I have classified amino acids, a simple classification of amino acids I have done. So the link for that video is there in the description below, and also it is appearing in the upper right corner right now. So in this video, let me explain you the most important points for some of the selected amino acids, and these are the high yield things for your all kinds of exam that you are going to take. So let's get into that. So the amino acids with high yield points for exam and on important amino acids, I have selected a few amino acids here. So our very first amino acid is proline. Now the proline is, as you can see, it has got alpha carbon here. It has got a primary primary carboxyl group, and it has got its amino group here, which is attached to the alpha carbon, is making a interaction with the side chain of the proline. See, the side chain of proline is CH2, 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 and that is making a linkage with the amino group here, which is attached to the alpha carbon, making a 5-membered rigid ring here. And this 5-membered rigid ring is the one that will make proline a special amino acid. And also proline is referred as, and it's an amino acid because all other amino acids amino group is always free whenever they are in free form, whereas this particular amino acid amino group is not free. It is making a bond with the side chain here. That is why this amino group, which is attached to the alpha carbon, it is called as a secondary amino group. So that means proline do not have primary amino group. So proline has got primary carboxyl group, but it do not have primary amino group. It has got a secondary amino group. This is the only amino acid out of 20 standard amino acid which has got a secondary amino group, and because of this speciality, it is also referred as a amino acid. So it has got secondary amino group there, and because this secondary amino group, it has got a 5-membered rigid ring. So whenever it is being part of a polypeptide chain, it leads to bends or kinks in a polypeptide chain, and that is why this proline is referred as a helix breaker, because it doesn't allow alpha helix to continue in a polypeptide chain, because it creates a bends and loops and turns in a polypeptide chain. So this is a speciality about proline there. Our next amino acid is glycine. Glycine is the most simplest amino acid because the side chain of glycine is just H, and also it's a precursor. Glycine is a precursor for heme, purine bases and creatine. So the heme is basically, it is a combination of glycine and succinyl CoA. That's how the heme is formed initially. That's how it starts. So glycine is a precursor for heme, it is a precursor for purine bases, that is adenine and guanine, and also it is a precursor for creatine. So because when the heme synthesis happens, it begins as glycine condensing with succinyl CoA. Purine bases adenine and guanine needs this glycine, and also in creatine synthesis, to make creatine phosphate in the skeletal muscle, you need glycine. So that's where importance of glycine comes in. So and also, as you know, glycine, it is part of collagen molecule. So in a collagen molecule, collagen has got the sequence glycine XY sequence. That means every first amino acid is glycine, then we have X and Y other amino acid, then you have glycine. So everywhere glycine comes in. So the glycine is frequently occurring amino acid in a collagen molecule. And also glycine can act as an inhibitory neurotransmitter in the spinal cord. And glycine is recycled in a peroxisome. So there is a recycling of glycine in a peroxisome done by an enzyme, alanine glycine transaminase enzyme, which is referred as AGT. Deficiency of alanine glycine transaminase enzyme will give rise to a disease, congenital disorder called primary hyperoxaluria. Because when the glycine is not recycled, so glyoxalate will be converted to oxalate, and oxalate levels will increase in the blood. Oxalate can combine with calcium to make calcium oxalate stones in the bladder, in the kidney, and it can give rise to renal failure. So these are some of the important points about glycine. Our next important amino acid is branched chain amino acids. We have leucine, isoleucine and valine. So the leucine, isoleucine, valine, one thing that is common in leucine, isoleucine and valine, all three of them are having a branches in a side chain. That's why they are called as branched chain amino acid. You can remember them as LIV, L-I-V. leucine, isoleucine and valine. What is the importance of this? Leucine, isoleucine, valine are highly consumed by skeletal muscles. Skeletal muscles are the ones which needs a lot of leucine, isoleucine and valine, and that's why if you look at protein supplementation in bodybuilders who take protein supplementation, they specially mention what is the concentration of branched chain amino acid there, because skeletal muscle proteins needs a lot of branched chain amino acids. So, and also these branched chain amino acids, they will, when they will undergo metabolism, so especially catabolism, there is a requirement of an enzyme called branched chain alpha-keto acid dehydrogenase complex. This branched chain alpha-keto acid dehydrogenase complex, it is allowing this branched chain amino acids to get into catabolism and give energy. So, whenever there is a deficiency of this enzyme, that will give rise to a disease called maple syrup urine disease, and the maple syrup urine disease has got severe neurological signs, and also characteristic odor of body fluids in this particular disease is a burnt sugar odor or maple syrup odor. Okay, remember that. So, these are some of the points related to branched chain amino acids. Our next amino acid is phenylalanine. Phenylalanine as an essential amino acid, it undergoes metabolism, like phenylalanine will be converted to tyrosine. That's why tyrosine is a semi-essential amino acid. So, phenylalanine conversion into tyrosine needs an enzyme called phenylalanine hydroxylase enzyme, and the deficiency of phenylalanine hydroxylase enzyme will give rise to a disease called phenylketonuria. What happens in this disease? Phenylketonuria patients will have mental retardation, they will have neurological issues, they can be hypopigmentation there, and most importantly phenylketone derivatives, phenylalanine derivative, phenylketones will be elevated in the body fluids, and one of the phenylketone is phenyl acetate, and this phenyl acetate has got a characteristic mousy or musty odor to body fluids. That's one of the thing that you got to remember there. And also, while conversion of phenylalanine into tyrosine, there is a requirement of a coenzyme called tetrahydrobiopterin, which is referred as BH4, and this tetrahydrobiopterin will undergo recycling process done by dihydropteridine reductase enzyme. If this particular enzyme is mutated, so that means there will be deficiency of tetrahydrobiopterin, and that will give rise to a disease called non-classic phenylketonuria, which is a malignant hyperphenylalanemia. That's the name of the disease, malignant hyperphenylalanemia, which is part of non-classic phenylketonuria. It is a more severe disease compared to classic phenylketonuria, because in this particular disease, the phenylalanine derivative, that is tyrosine, which is giving to dopamine, norepinephrine, epinephrine, they all will be decreased. Tryptophan conversion into serotonin needs tetrahydrobiopterin, that is also decreased, serotonin will be decreased. Conversion of arginine into nitric oxide is needs tetrahydrobiopterin, so that will be decreased. So, overall in this particular disease, there will be decrease in dopamine, norepinephrine, epinephrine, serotonin, and there will be increase in and also decrease in nitric oxide, and there will be increase in prolactin, because normally dopamine has an inhibitory effect on prolactin secretion. When the dopamine is decreased in this disease, prolactin level will be increased. So, these are some of the points for phenylalanine. Now, let us move on to see what is the importance of tyrosine. Now, tyrosine is a derivative of phenylalanine. So, phenylalanine hydroxylase is the one that gives tyrosine. So, once you get tyrosine, so tyrosine participate in protein synthesis, and tyrosine is a semi-essential amino acid. So, that means it is needed only under positive nitrogen balance conditions. So, tyrosine actually it will undergo when it will give rise to special products. Tyrosine can give rise to special products like catecholamines. We have three catecholamines: dopamine, norepinephrine, epinephrine. Not only that, tyrosine is converted into melanin in melanocytes. Tyrosine is diverted into melanin synthesis in the skin cells called melanocytes. Tyrosine can also give T3 and T4, thyroid hormone. So, these are special products that come from tyrosine. And in the tyrosine catabolism, when the tyrosine is broken down or catabolized to give energy, we need certain enzymes there. So, one of the enzyme that is required in the catabolism of tyrosine is a homozentisate oxidase enzyme. Deficiency of homosynthesis oxidase will give rise to a disease called alpha-keto, and this alpha-keto patients will show a hyperpigmented, like brown to blackish pigmentation in cartilaginous tissue, and that particular thing we call it as ochronosis. And the ochronosis is the pigmentation of cartilaginous tissue, brownish to black pigment, and this happens because in this particular disease there is accumulation of homosynthesis in cells, and the homosynthesis will auto-oxidize into quinone derivative, which has got higher affinity for cartilaginous tissue, and the quinone has got brown to black pigment, and quinone wherever it is present, it will have that will basically discolor the tissue including the body fluids. That means urine will have a brown to blackish pigmentation in alpha-keto patients. Tyrosinemia. So, there is a type 1 tyrosinemia which is also called as tyrosinosis because of the deficiency of another tyrosine catabolizing enzyme called fumarate acetoacetate hydrolase enzyme. Deficiency of this enzyme will lead to elevation of fumarate acetoacetate, and that fumarate acetoacetate will be converted into succinyl acetone, and succinyl acetone it actually is a DNA alkylating agent, and that causes tumor formation. So, and also fumarate acetoacetate, it has got a characteristic odor which is referred as cabbage like body odor. So, patients with tyrosinosis will exhibit cabbage like body odor. And then the tyrosine which is going into melanin formation in the melanocytes, that will be done by copper dependent tyrosinase enzyme, and if there is a deficiency of copper dependent tyrosinase enzyme, that will decrease melanin synthesis in the melanocytes, and that will give rise to generalized hypopigmentation in these patients, and the name of the disease here, it is referred as albinism. In albinism patients, they will have generalized hypopigmentation, they do not have mental retardation, they do not have any problem with the IQ, and they will have photophobia, and they will have high risk for skin cancers because melanin protection is not there for them. So, our next amino acid is tryptophan. Now, the tryptophan, it is derivatives for tryptophan. So, it is serotonin, melatonin and niacin. These are the special products that come from tryptophan. Serotonin is there in serotonergic neurons, melatonin is synthesized in pineal gland, and niacin is synthesized in most of the tissues using tryptophan. 60 milligram of tryptophan is converted into 1 milligram of niacin, and that needs certain vitamins like B2, B6, and also it needs iron for that conversion. So, carcinoid syndrome patients, they will in these patients what will happen? So, there will be excess diversion of tryptophan into serotonin formation rather than niacin formation. That is why carcinoid syndrome patients, they will have high serotonin levels and they will have less niacin level, give rise to pellagra signs and symptoms. The other disease called Hartnup disease. In Hartnup disease, there is autosomal, this is an autosomal recessive condition where there is a decrease in the absorption of tryptophan in the intestine and decrease in the reabsorption of tryptophan in the renal tubules, overall giving rise to tryptophan deficiency and thereby giving rise to niacin deficiency. So, now let us move on to see glutamate. Glutamate, it is a component of antioxidant glutathione. You know, glutathione is a antioxidant, especially like in the red blood cell. Glutathione is gamma glutamyl cystinyl glycine. So, that is where is the glutamate is there, right. And now other than that, so glutamate is an excitatory neurotransmitter in the central nervous system. Glutamate can undergo decarboxylation into GABA, gamma aminobutyric acid, and which is an inhibitory neurotransmitter in the central nervous system. Coming to methionine. Methionine, it is again an essential amino acid and it is part of our polypeptide chain. Methionine is formed as a special product called S-adenosyl methionine, and this S-adenosyl methionine, it is participating in the methylation reaction. It is a methyl group donor. So, the methylation reactions are needed in creatine synthesis, epinephrine synthesis, means conversion of norepinephrine into epinephrine, melatonin synthesis, DNA methylation process, and also lecithin formation. So, like phosphatidylcholine, that is what is lecithin. So, that means very important molecular formations in our body needs S-adenosyl methionine, which is coming from methionine. Now, deficiency of deficiency of an enzyme called cystathionine beta synthase in methionine metabolism. Methionine is converted into acetinosyl methionine. Once it participates in methylation reaction, so it will give rise to homocysteine. Homocysteine can go back into methionine or it can go into cysteine formation. That is why cysteine is a semi-essential amino acid. So, there is an enzyme called cystathionine beta synthase. Deficiency of this enzyme will give rise to a disease called classic homocysteinemia, and in this type 1 homocysteinemia cases, there will be increase in homocysteine and also there will be increase in methionine. Deficiency of folate and cobalamin, these are the two vitamins, are needed in the recycling of methionine from homocysteine. So, if there is any deficiency of these two vitamins, cobalamin and folate, they will lead to decrease in methionine formation, thereby decrease in S-adenosyl methionine formation. Our next amino acid is cysteine. Cysteine is a sulfur containing amino acid. It has got a thiol group, that is a SH group which is free there, and that SH group, it can interact with the neighboring cysteine or another free cysteine molecule or cysteine in polypeptide chain and make a disulfide linkase, and this disulfide linkase so if there are two free cysteine molecules, if they come together and make a disulfide linkase, and that entire molecule we call it as cysteine. And there is a disease called cystinuria. In this disease, cysteine is just going out of our body in the urine. There will be loss of cysteine. So, loss of one cysteine means there is loss of two cysteine molecules. Cysteine is an oxidized form of cysteine. One cysteine has two cysteine. So, if there is a loss of one cysteine, there is a loss of two cysteine molecules. So, that is about cystinuria there. And also disulfide linkage in a polypeptide chain helps in tertiary structure formation. It is kind of stabilizing the tertiary structure, remember that. And our next amino acid is arginine. Arginine is a precursor for urea, creatinine, nitric oxide, which is a vasodilator, which is a neurotransmitter, and also it is a part of augmentin, which is an anti-hypertensive, and ornithine, which is a polyamine, which is part of our DNA. So, basically, so means all the things stones, polyamines are rich in histone formation. L-arginine is used in the treatment of angina, hypertension, erectile dysfunction, because arginine is converted to nitric oxide, has got that vasodilatory function. Asymmetric dimethyl arginine is a close relative of arginine, is a marker of vascular disease. Arginine is a marker of good endothelial function, whereas asymmetric dimethyl arginine ADMA is a marker of vascular disease. We have serine, threonine and tyrosine. These are hydroxyl containing amino acids which are participating in post-translational modification of glycoproteins, O-linked glycoproteins and N-linked glycoprotein synthesis. Asparagine, it is participating in N-linked glycoprotein and proteoglycan synthesis, and there is a role of dolicol phosphate in N-linked glycosylation process. Coming to glutamine. Glutamine, it is a donor of amino group in a cellular metabolism. Wherever you see amino group containing metabolic intermediates like N-acetyl glutamate, N-acetyl galactosamine, N-acetyl glucosamine, glucosamine, galactosamine, all these things, wherever amino group is part of a metabolic intermediate, remember that is coming from glutamine. And also glutamine, it helps in transport of amino group from peripheral tissues to the liver for detoxification process, and glutamine can be used as energy source for tissues like intestine. And also L-glutamine supplementation called Endari is an FDA approved treatment for sickle cell anemia patients, because L-glutamate, it replenishes glutathione synthesis in red blood cells. So, thereby it will prevent oxidation of their red blood cell membrane, thereby decreases hemolytic process there. So, glutamine levels can be elevated in urea cycle disorders and any conditions that leads to hyperammonemia, because glutamine is the one that is basically coming from alpha-keto glutarate combining with ammonia to make glutamate, and glutamate takes one more ammonia to make glutamine. So, whenever there is excess glutamine, it simply indicates that there is hyperammonemia going on there in the body. So, our next and last amino acid is histidine. Histidine has got this imidazole group here, which has got a pKa7 when it is part of a polypeptide chain, and this pKa7 is very near to physiological pH 7.4. That means 50% of the histidine is protonated, 50% is deprotonated. That is why histidine can act as a physiological buffer in our polypeptide chain. So, the buffering capacity of histidine, it comes from protein, it will come from histidine. So, that is about information about selected amino acids, important or high yield points about selected amino acids. I hope this video has helped you in understanding these two videos, part 1 about classification and part 2 about high yield points about selected amino acids. I hope this video has helped you in understanding about amino acids and to do well in the exams or whatever the competitive exams that you are taking. So, if you haven't subscribed to my channel, please subscribe to this channel so that you get regular updates, and also don't forget to click that bell icon to get notification. So, I will see you in my next video. Till then, you take care. Thank you.